Hello dear engineers, the subject of this paper is fatty liver level recognition using PSO image segmentation and analysis. As an introduction, fatty liver or liver hepatic glycogen is one of the most common disorders of the liver nowadays. Clinical detection of this disorder by a human expert is increasing. That's due to our lifestyle, which leads us toward this phenomenon. Accumulation of extra fat from food or alcohol in liver could cause fatty liver disorder. If it caused by heavy use of alcohol, then it is called alcohol fatty liver disorder or AFLD. If it caused by food, then it is called non-alcoholic fatty liver disorder or NAFLD. This disease is widespread nowadays due to overuse of food and alcohol and less activity among people, so extra fat doesn't burn enough and adds up to liver. Fatty liver normally doesn't show any symptom in early stages. If it continues without treatment, it may cause cirrhosis or liver cancer. As it has fatal effect, research to find the proper solution is essential. Fatty liver shows signs of tiredness and pain in upper right side of the chest, which then it is time to visit the doctor. The doctor first asks the patient about symptoms and then prescribe, and in a severe situation, to the blood test. The problem is sometimes blood test fails to detect NAFLD and ultrasound scan is needed for robust detection. So making a fast and accurate expert system for fatty liver detection is essential in each clinic. That's why we intended to make one. We recognize its label by 4 layers of markers using macroscopic image data. It is possible to use artificial intelligence techniques such as machine learning, pattern recognition, image processing, and evolutionary computation to make an expert system for fatty liver detection. Proposed expert system works based on variety of image processing techniques and algorithms to detect fatty liver and recognize its level by 4 markers. Main privilege of expert systems are their lack of error and tiredness comparing with human expert. For segmentation technique of watershed, K-means, and PSO segmentation are employed to determine disorder level. Performance metrics of accuracy, F-score, and IOU or Jacquard for evaluation the precision of the proposed method is used. Finally, fatty liver level is calculated based on the amount of fat deposit inside segmented image. Experiments are conducted on multiple data sampled in high resolution with microscope zoom bigger or equal of 200, which are collected from the internet. All performance metrics and comparison return satisfactory result in comparing with traditional methods. Proposed system could achieve average accuracy of 0.922 for all samples comparing with ground truth data. Additionally, F-score and IU performance metric return value of 0.872 and 0.907 respectively. One of the mentionable types of research in fatty liver detection using image segmentation is belong to Gao and partners in 2019. They used masked RCNN algorithm to segment fat deposit in liver samples and achieved 78% average accuracy with their method. Another research belonged to Zhang and partners in 2021. They used region of interest ROI on magnetic resonance imaging or MRI in order to segment liver images for NAFLD and received promising result. Another method is belonged to Goldberg in 2017. They used computed tomography or CT images belong to 107 subjects in 3 slices to estimate fat deposit in patients by common segmentation techniques and achieved high correlation of 0.930 for their analysis. Rao and partners in 2021 present a fully automatic liver statosis prediction model using 3 deep neural network and semantic segmentation to achieve 99% accuracy for their system. Native and partners made a system for fatty liver detection using segmentation technique in 2014. They used 54 images from 9 subjects and could achieve 99.3% accuracy in fatty liver detection by their system. Significant research to characterize normal fatty and heterogeneous liver using textural analysis of liver ultrasonic images introduced by Jim Hare and partners in 2015. Their system selects region of interest by segmentation and uses wavelet transform to extract statistical features and discriminates the type of diseases in liver, including fatty liver. The need for strong single image based segmentation is vivid, which need to be satisfied. At the beginning, for the purpose method, at the beginning the liver image investigates and analyzes by human expert and sent to the computer system for automatic recognition. Some pre-processing steps such as intensity adjustment, histogram equalization, and canny edge detection applies on input image for improvement and better recognition process. There are 4 segmentation techniques, namely also K-means, watershed, and PSO. The novelty of this research lies behind using a nature-inspired algorithm for segmentation on single images, which outperform traditional single-based methods. We didn't compare purpose method with deep learning methods, as our method is single-based and deep learning is multi-image based. In order to evaluate the performance of segmentation, 3 performance metrics of accuracy, F-score, and IOU are employed. Final step is consisted of fatty liver level recognition based on fat deposit amount and markers amount and marker selection process. As we can see here, there are 4 markers of 1, 2, 3, and 4. 1 is healthy, 2 is very unhealthy, 3 is medium level unhealthy, and 4 is just a little bit unhealthy. Here on the left we can see some sample employed inside the experiment, and on the right we can see in each row different blood samples of the liver in different stages: healthy liver, liver in early cirrhosis, and in the developed cirrhosis. Here we can see the flowchart of the proposed method. It starts by inputting data, analyzing by human expert, sending for calculation, pre-processing, then for segmentation by 4 techniques, and finally it's going to be evaluated, and fat deposit or fatty liver level will be determined. PSO is a nature-inspired mathematical algorithm based on natural behavior of fish and birds in the soil. Group of animals move toward more optimal position in the space, which leads to optimal or better solution. The quality of the movement is based on number of iteration and population size. In PSO segmentation, image matrix converts into a vector and cluster by basic K-means clustering. The process improves zeroing time based on number of segments, number of initial population, and iterations. Finally, vector segmented data convert back into matrix from with different cluster, which are our threshold level or segments. Each segments represent by a color, which is final segmentation image. At the right we can see the pseudocode of the PSO segmentation. Here we can see different markers. Marker 1 is blue at the left, A. B is marker 2, red. Marker 3 is yellow, which is C, and D is marker 4 or black spots. You can see the result of the marker 2 is 21% of fat. Here at the left we can see a sample which is segmented by different methods. First it's edge detected and adjust intensity happened on it. Then with also watershed, K-means, and PSO segmentation with 2 clusters is segmented. At the right we can see another experiment result with PSO and boundaries around the deposits or fat deposits. Here we can see an image with 4 markers of a fatty fatty liver sample. The fat area of the marker is at 22.6%. The area for the marker 2 is 21. The area for the marker 3 is 43, and the area for the marker 4 is 12. Now, if the marker 2, 3, and 4 are hepatic glycogen or fatty, the area of hepatic glycogen is 62%, which marker 1 is healthy, so it doesn't count. Marker 2 is fully hepatic glycogen, which we consider it all of it, or 1. Marker 3 is 80% fatty, we consider it 0.8, and marker 4 is just half of it going to be considered. So the area is going to be calculated based on the fat deposit multiplied by its level, which is going to be 62.89% for this sample. Here we can see another example of fatty liver. A cell at the top left we can see original image, ground truth, and finally PSO segmented. And the second row at the left we can see the result on BW image or black and white image, then on the original image, and finally at the bottom right we can see how the cost function worked for the PSO segmentation. In the evaluation section, K-means algorithm could cluster closed groups of samples into different number of clusters based on their distance in the space. Same method could be employed for image pixel value, which could be gray level, intensity, or color. Obviously, this method is categorized into cluster-based segmentation methods. Another example is watershed image segmentation. It is a region-based method, and this algorithm is based on draining in basins or rivers. It works based on water level, as water height represent the separating line between one region or segment to another. A commonly used segmentation technique is called also thresholding segmentation. It categorized as a threshold-based segmentation. It works as a thresholding process for pixel intensity value in order to minimizing inter-class variance for separating pixels into two classes of foreground and background. Number of threshold level determines the number of segments here. In comparing a return image versus ground truth by considering a white color or 1 as a positive and black color or 0 as a negative in a black and white image, then we have TP is when the model correctly predicts the positive class. It is true positive. True negative or TN is when the model correctly predicts the negative class. False positive or FP is when the model incorrectly predicts the positive class, and false negative is when the model incorrectly predicts the negative class. Here we can see in this figure samples for two classes, true positive, false positive, true negative, and false negative on a table, and we can see how they are going to calculate it as we need it for the evaluation section. In order to evaluate the method, three performance metrics of accuracy, F1 score, and intersection over union or IOU are employed. Accuracy simply is the ratio of correctly classified pixels for the object, which we can see is TP plus TN divided by TP plus TN plus FP plus FN. Then we need precision and recall for the next evaluation metric. Precision is TP divided by TP plus FP, and recall is TP divided by TP plus FN. So here the boundary F1 contour matching the score indicates how well the predicted boundary of each class aligns with the true boundary. F score is 2 multiplied by precision multiplied by recall divided by recall plus precision. Intersection over union, also known as the Jacquard similarity coefficient, is the most commonly used metric for segmentation. IOU is a statistical accuracy measurement that penalizes the false positives. For each class of IOU is the ratio of correctly classified pixels to the total number of ground truth and predicted pixels in the class, and it is calculated by TP divided by TP plus FP plus FP. Here we can see a result related for all the samples for accuracy performance metrics and all the algorithms of OTSO, watershed, k-means, and PSO. We can see 1 to 4 samples are healthy, 5 to 8 are early zeros, and 9 to 12 are developed zeros. As we can see, for the accuracy, PSO segmentation had better performance than others, except one sample number 6, which also had outperformed others. For F-score, we have the same situation, just also had 2 samples of 5 and 8 in comparing with ground truth data, and in the other samples, PSO outperformed other methods. For IOU or Jaccard performance metrics, we have also for 2 samples of 5 and 8, which outperformed others, but in the other samples PSO outperformed them. Here we can see the result for accuracy, F-score, and OU from left to right, and for different algorithms as a box plot. As we can see, PSO always is closer to 1 than other samples. Also here, after segmentation and edge detection, we can calculate the fat deposits for the samples. Here we consider just 2 of them, a healthy and an unhealthy. Top row is a healthy one, which is fat deposit of 2.67 percentage, and the second row is the unhealthy one, which is detected, and fat deposit is 21%. As a conclusion, by employing nature-inspired algorithm in image processing and especially image segmentation, it is possible to achieve even more precise results from segmentation. This research proved that PSO algorithm could reach to that point, increasing fatty liver level recognition accuracy and speed, a perfect replacement for human expert, and better performance in OTSO and PSO segmentation techniques are returned mostly by PSO. Even by changing the hyperparameter of traditional methods, PSO segmentation returned superiority result over all of them. It can be concluded that proposed method could be used in both AFLD and NAFLD diseases. Using other nature-inspired algorithms such as genetic algorithm and others in segmentation techniques of future works, it is suggested to employ proposed method on different data with higher or lower microscopic zoom. Here we can see our references which are used for this research. There are 31 of them, and thank you so much for your attention.